Right. A warm welcome again. So we'll start this second session today. Uh, so we are going to discuss about the eukaryotic intracellular organelles one by one. So in our earlier session, we discussed about differences between prokaryotes and eukaryotes, then followed by what are the components of a prokaryotic cell, and also we have seen what are the components of a eukaryotic cell, right? So we have seen so many internal organelles present in the eukaryotic cell. So we are going to go and discuss one by one about each type of organelles. We will be majorly focusing on the structure of intracellular, eukaryotic intracellular or organelle and the functions that are carried out inside the cell, right? So in the session 2, we are going to focus on mitochondria, Golgi complex and endoplasmic reticulum. ER stands for endoplasmic reticulum. Right. So we can see mitochondria here. So there are several thousands of mitochondria inside a cell, and we all know the major function of mitochondria is to produce ATP, adenosine triphosphate. So that's why this mitochondria is also called as the powerhouse of the cell. Okay, so why it is called as powerhouse of the cell? Because it has the ability to synthesize the energy molecules that are required by the cells. So the energy molecules inside the cell will be in the form of ATP, adenosine triphosphate. Since it has the ability to synthesize this ATP, that's why it's called as the powerhouse of the cell. All the ATP that is there inside the cell is synthesized by this particular organelle called mitochondria. So how does this mitochondria structurally look like? So this is mitochondria is a double membrane bound intracellular organelle. That means it has two membranes on its envelope, the outer membrane and inner membrane. Okay, so the inner membrane is again invaginated into several folds called as cristae. So cristae are the invaginations present inside the inner membrane of the mitochondria, and this foldings plays very crucial role in several enzymatic reactions as well as several reactions that's happening in the electron transport chain, right? So many varieties of metabolic pathways happens inside the mitochondria, starting from your Krebs cycle or oxidative phosphorylation or electron transport chain. These are all the metabolic pathways happens inside the mitochondria itself, right? So in order to perform or carry out all these metabolic pathways, the mitochondria should have specialized enzymes in it. So that's why mitochondria has ribosomes again inside the mitochondrial structures. We all know ribosomes are the organelles which helps in the protein synthesis. Once proteins are synthesized, they'll be converted into an enzyme, and then this enzyme will be helpful in carrying out the metabolic pathways. So here you have so many number of ribosomes inside the mitochondria, as well as mitochondria has a DNA inside it, which is called as maternal DNA or mitochondrial DNA. So DNA present inside the mitochondria is called as mitochondrial DNA, and this mitochondrial DNA has very very high significance, because it is a hereditary structure which came from maternal origin. Okay, that means it's been transferred from mother side to the child side, not from the father side. Why? Because this when there is a fusion between egg and sperm, you have the mitochondria which is there inside the egg or the ova, but you do not have any mitochondria inside the sperm. Even though there are mitochondria, but only the genetic material will be transferred or going into the ova, but not the mitochondria. Mitochondria, what you see in the progeny, is coming from the mitochondria that is from maternal origin. Okay, so that is the significance of mitochondrial DNA, which is highly conserved from species to species, and it is having maternal origin. And you have the cristae, and you have the ATP synthesis centers, where ATP synthesis happens in the folds of the cristae in the electron transport chain. So let's have a look at what are the major functions of mitochondria. So first major function of mitochondria is ATP production. As we are discussing, a mitochondria is called as powerhouse of the cell and has the capability to produce the ATP. How ATP is being produced? ATP is being produced by carrying out several metabolic pathways inside the mitochondria. Okay. So cellular respiration, another major function of mitochondria. Cellular respiration in the sense the breaking down of complex carbohydrates into simple forms of sugars, which can be converted into energy molecules, is called cellular respiration. The enzymes involved in the cellular respiration are synthesized inside the mitochondria. And the third major function of mitochondria is metabolism and biosynthesis, right? So mitochondria, as we are discussing, has so many enzymes in it, and this carry out metabolisms like your Krebs cycle or citric acid cycle, and electron transport chain are carried out inside the mitochondrial matrix. So this is the matrix, and these are the invaginations called cristae. So inside the matrix, so many metabolic pathways are being carried out in the matrix, and which are again helped by the enzymes synthesized by the mitochondria. And biosynthesis. So mitochondria also helps in synthesizing heme and other hemoglobin components of the cell. So that's also one major importance of mitochondria. Apoptotic regulation. So here, apoptosis in the sense a programmed cell death mechanism. So when cell needs to kill itself, right? When cell needs to destroy itself, what cell will do is a cell will activate a specialized enzymes called caspases. These caspases will trigger and perform the cell program called programmed cell death. Okay, so caspase 3, caspase 9, and so many other caspases are involved in the programmed cell death mechanism, that is apoptosis of the cell. So regulation of the apoptosis is done by the mitochondria. Okay, so mitochondria are going to activate the caspase enzymes and helps in regulating the apoptotic pathway. So that's also one major importance of mitochondria. And another function which is not mentioned here of mitochondria is the synthesizing of ROS or reactive oxygen species. So ROS or reactive oxygen species are synthesized by mitochondria. These are not good to the cells. Okay, why they are being synthesized? Because since there are so many metabolic pathways are happening, and even the final electron chain acceptor is an oxygen molecule. Since oxygen is able to get the final electrons, it will be oxygen molecules will be again activated. They will be converted into so many active forms like reactive oxygen species. Reactive oxygen species are nothing but highly reactive oxygen molecules which has the ability to go and react with so many wide varieties of biomolecules present inside the cell, right? So if there is reactive oxygen molecule inside the cell, it may go and react with the protein, it may go and form a bonding with the carbohydrate, it may go and attack a nuclear genetic material. So many things can happen. That's why a reactive oxygen species concentration should be minimal inside the cell. Then only cell will be cell can survive for longer period of time. If the ROS content or ROS levels are high, it may even leads to the triggering of cancers. That's why we should always minimize the ROS levels present inside the cell. But here in case of mitochondria, which is able to synthesize ROS, even though it is not good for the cell, but cell has its own mechanism to curb the reactive oxygen species generated by mitochondria. We will discuss more about that in coming sessions. So that's about mitochondria. So mitochondria structure look like this, a bean-shaped structure, and it has two membranes, outer membrane and inner membrane. Inner membrane is folded into cristae, and it has a matrix where metabolic pathways will happen. What metabolic pathways happens inside the matrix? Mostly your Krebs cycle and electron transport chain will take place inside the matrix, and all the enzymes involved in the metabolic pathways are synthesized by the ribosomes, again present inside the mitochondria itself. And major functions: ATP production, cellular respiration, also called as oxidative phosphorylation, metabolism, metabolic pathways taking out inside the cell, biosynthesis of heme and other biomolecules, apoptotic regulation, regulation of programmed cell death mechanism by the mitochondria, and the last one is production of ROS species. So these are some of the major functions of mitochondria. So let's move on to the next organelle. So we have what you see here is called as Golgi complex. So this is again a double membrane bound organelle which has again two membranes. This is a sac-like structure which has two ends, a front end and a back end. Front end is called cis phase and the back end is called a trans phase. Okay, so this Golgi complex will be always connected with endoplasmic reticulum. We have seen the structure, how endoplasmic reticulum look like. It is a net-like structure which is projecting from the nuclear membrane, right? And on the endoplasmic reticulum, we can see the ribosomes present, and proteins will be synthesized on top of the endoplasmic reticulum. These proteins will move into the endoplasmic reticulum again, and they will be post-translational modifications happening inside the endoplasmic reticulum. And these post-translated proteins, what they do is they will be packed inside vesicles. Vesicles are small sacs or small round balls like this. This is incoming vesicles from endoplasmic reticulum, and then they will be moving on to the Golgi complex. Okay, so inside the Golgi complex, there will be sorting of the proteins. All the similar type of proteins will be sorted into one bag or one sac, labeled, and then they will be transported to a particular region of the cell or to the exterior of the cell. Okay, so major function of Golgi complex is sorting of the proteins and enzymes which are coming from the endoplasmic reticulum and directing them to proper destinations. The destination can be other intracellular organelle, or it could be going out of the cell by exocytosis. A process called exocytosis helps in making the vesicle sac go and attach to the cell membrane, and these cell membrane on the membrane of the vesicle will fuses, and the contents will be released out of the cell, right? So coming to the structure, it has two ends, as I said, cis phase and trans phase. Cis phase will be always projecting towards the endoplasmic reticulum, and trans phase will be always projecting towards the cell membrane. So here, incoming vesicles are nothing but small bags or sacs loaded with proteins and other biomolecules, comes to these sacs or Golgi complex. They will be segregated, sorted inside this structure, and again repacked into vesicles, and then they will be transported into transported to different parts of the cell. So these are incoming transport vesicles merging with the Golgi complex, getting sorted inside the Golgi complex, and then these are outgoing vesicles from the trans end, where these are newly formed and only similar types of proteins and molecules will be packed in one bag and labeled with a specific tag, carbohydrate tag, so that it will move to a proper destination. Okay, so tagging, sorting, and directing are the major functions of the Golgi complex. So second major function in plants, Golgi complex or Golgi apparatus is involved in the secretion of cell wall components like cellulose, semicellulose, lignin, as well as lipids and glycoproteins which are present in the cell membrane. All these components are secreted and transported by the Golgi complex. Next, in case of animal cells, your mucus proteins, lactoproteins, collagen, and melanin, these are all the molecules or proteins which are transported, sorted, segregated, and transported to different regions by the Golgi apparatus. Coming to the next major function, formation of the cell membrane. If at all cell need to undergo cell division and the cell membrane has to be formed, then again Golgi complex plays a crucial role in taking the lipoproteins and other proteins from the endoplasmic reticulum and helping in formation of the new cell membrane. Right? So last major function is involved in the formation of cellular organelles. Right? So intracellular organelles like lysosomes, which are membrane bound organelles, and spermatozoa as well as plasma membrane, these are all organelles, intracellular organelles, which are synthesized with the help of the Golgi complex transport mechanism. So these are the major functions of Golgi complex. So let's move on to the next organelle, that is endoplasmic reticulum, right? So endoplasmic reticulum, the name itself says that it is a reticular structure or a net-like structure which is extending from the nuclear membrane. So this is the nucleus and this is the endoplasmic reticulum, which is in continuation with the nuclear membrane. So I'll tell you why this endoplasmic reticulum has to be in continuation with the nuclear membrane. So we all know that nucleus has the genetic material, right, in the form of DNA. So when the protein synthesis has to happen, DNA has to be converted into RNA, specific type of RNA called mRNA or messenger RNA. So this messenger RNA has the message for synthesizing a particular protein. A particular type of a gene will be translated into a particular type of protein. In between these two, we have the messenger RNA. Okay, so messenger RNA is produced inside the nucleus, and messenger RNA will be transported from the nucleus through the nuclear membrane into the endoplasmic reticulum surface, since there is a continuation between nuclear membrane and endoplasmic reticulum membrane. So protein synthesis always happens inside the cytoplasm. Protein synthesis will not happen inside the nucleus, right? So mRNA, which is formed by transcription of the DNA, will be moved towards the cytoplasm and it will go and bind on to the ribosomes which are present on the surface. So when an mRNA attaches to the ribosome, then translation happens. Okay, here in case of DNA, DNA to mRNA is called as transcription, and transcribed mRNA will be moving out of the nucleus to the cytoplasm, where it gets translated into proteins. Transcription, translation. Okay, so So, so that's the importance of continuation of nuclear membrane and endoplasmic reticulum membrane. And this is called a rough ER because ribosomes are studded on top of the endoplasmic reticulum, and this structure is called as smooth ER, which does not have any ribosomes on its top. So let's have a look at what are the major functions of rough endoplasmic reticulum and smooth endoplasmic reticulum. So first major function of rough endoplasmic reticulum is it provides a surface for binding of the ribosomes for protein synthesis to happen. Okay, mRNA comes from the nucleus, the mRNA will go and attach to the ribosomes. These ribosomes will be bound onto the rough endoplasmic reticulum, and after latching onto the rough ER, then only protein synthesis will start. Second one, formation of glycoproteins, right? So another very important function of rough endoplasmic reticulum is post-translational modifications. So just now we discussed about what is transcription and what is translation. In case of nucleus, the genetic material in the form of DNA will be converted into mRNA by transcription, and once mRNA is produced inside the nucleus, it will be transported out to the cell, and inside the cytoplasm they will go, and this mRNA binds to the ribosomes, and mRNA will be converted into proteins based on the codon sequences present on the mRNA. Okay, amino acids specific for a particular codon come and attach to the ribosome, and the next codon call for another amino acid, and between these two amino acids there will be a peptide bond formed. So what you get is a polypeptide chain having several different types of amino acids based on codon sequence of the mRNA. So this polypeptide chain is nothing but several amino acids with the linkage, a peptide bond between amino group and carboxyl groups of amino acid. But this is not a complete protein. If it needs to convert into complete protein, post-translational modifications needs to happen, like a glycosylation or any other post-translational modification happens inside the endoplasmic reticulum. So once the proteins are synthesized on top of the ribosomes outside the endoplasmic reticulum, protein will move into the endoplasmic reticulum, rough endoplasmic reticulum, and post-translational modifications happens inside the rough endoplasmic reticulum, and then it will make into a complete protein, and then they will be moved to the Golgi complex where they will be sorted out, and then they will be clubbed and be transported to the destination, right? So that's about formation of glycoproteins, which is one of the post-translated protein, and synthesis of precursors for lysosomal enzymes. There are so many varieties of enzymes present inside the lysosomes. All these precursors of these enzymes are synthesized here in case of rough endoplasmic reticulum, and formation of smooth endoplasmic reticulum is also one major function. So formation of smooth endoplasmic reticulum means this: simply take off all the ribosomes from the structure, and this specialized structure helps in biosynthesis of the lipids. So let's have a look at what are the major functions of smooth endoplasmic reticulum. So smooth endoplasmic reticulum majorly helps in biosynthesis of phospholipids, cholesterol, and hormones, as well as it helps in regulation of the lipids as well as calcium hemostasis. So I'll wind up this session here. I'll continue with the next set of internal organelles in the coming session. Thank you.